few major pathways used to break down sugars. With cellular respiration, the carbohydrate glucose is broken down all the way to carbon dioxide in water. The process begins with glycolysis, which splits the glucose in half. Fermentation, on the other hand, breaks down glucose, but it results in organic waste products. It also begins with glycolysis, but the products are not completely broken down as they are in cellular respiration. Cellular respiration begins with glycolysis. This is an anaerobic process because no oxygen is needed and occurs in the cytoplasm of most cells. It begins with 1 glucose and ends up with 2 pyruvates and a net gain of 2 ATP. The Krebs cycle is where we have complete oxidation of the substrate, resulting in 2 GTP, 2 FADH2s, 6 NADHs, and 6 carbon dioxide. ATP is then made through a series of redox reactions. The electron transport chain is where the redox reactions occur using a series of membrane-bound carrier molecules that pass electrons to a final electron acceptor. This creates a proton pump gradient used to make ATP. Glycolysis is also known as the Embden-Meyerhof pathway. It begins with the 6-carbon sugar glucose. In order to begin the breakdown process, we split an ATP to put some energy into the reaction and end up with glucose 6-phosphate. This is rearranged to fructose 6-phosphate, and a second ATP is split to create fructose 1,6-diphosphate. This is converted to dihydroxyacetone phosphate before splitting into 2 molecules of glyceraldehyde 3-phosphate, each with 3 carbons. With energy released as NADH, it is converted to 1,3-diphosphoglycerate. ATP is finally released as this is made into 3-phosphoglycerate, releasing 2 ATP, which essentially is paying back the energy required to get started. It is then rearranged to 2-phosphoglycerate and then to phosphoenolpyruvate. In the final step, we end up with 2 pyruvate and 2 ATP, giving the entire process a net gain of only 2 ATP. Here we begin with the glucose breaking down or adding this phosphate onto carbon number 6, it being rearranged, adding more phosphate on, the dihydroxyacetone phosphate splitting into the glyceraldehyde 3-phosphate, the first ATP being removed, with 2 of them being produced to repay the ones that were initially put in, some more rearrangement, and finally 2 ATP being added at the end. This is an animation here of glycolysis.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Prior to entering the Krebs cycle, A carbon is pulled off pyruvate and released as carbon dioxide. This leaves us with the 2-carbon acetyl-CoA, and energy in the form of NADH. This combines with the 4-carbon malate from the previous cycle, which is converted to oxaloacetate and then converted to the 6-carbon citrate. Citrate rearranges to isocitrate and then releases carbon as carbon dioxide, forming the 5-carbon alpha-ketoglutarate. This will release additional energy as NADH. Another carbon is removed as carbon dioxide from alpha-ketoglutarate to form the 4-carbon succinyl-CoA and release another NADH. In this process of rearranging succinyl-CoA to succinate, energy is released as GTP. The succinate is rearranged to form the 4-carbon fumarate and releases energy as FADH2. The fumarate rearranges to form the 4-carbon malate, releasing NADH. Finally, the malate is rearranged to form the 4-carbon oxaloacetate to repeat the cycle and release NADH. In each step, energy is released and a more stable arrangement is formed until it cycles back to take another acetyl-CoA. Each glucose forms 2 pyruvates needing 2 turns of the cycle. It is aerobic because oxygen is required to attach the carbons to create carbon dioxide. Here we have another animation of the Krebs cycle.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
There are some detours which can be made from glycolysis depending on the needs of the cell. One of these is the pentose phosphate pathway. This begins with glucose, but glucose 6-phosphate is used to create the 5-carbon sugars ribulose, xylulose, and ribose. The 5-carbon sugars are precursors to other anabolic pathways. One example is ribose is used to create the purine and pyrimidines of the nucleotides and to form the ribo in ribonucleic acid and deoxyribonucleic acid, or RNA and DNA. This only results in the net gain of 1 ATP. So you can see where it branches off here for the purine and pyrimidine metabolism. The Entner-Duodorf pathway is a substitute for glycolysis in some bacteria. It yields other precursor metabolites for the cell in only one ATP. The process catabolizes glucose to pyruvate like a glycolysis, but uses different enzymes and different intermediate steps. This is found in organisms such as Pseudomonas and Enterococcus faecalis. The electron transport chain uses energy from the electrons to pump protons across a membrane in the cristae of the mitochondria. The source of electrons is the NADH and FADH2 from the Krebs cycle. The membrane is embedded with carrier molecules, which will pass the electrons down the chain. So it can be seen here with the carrier molecules. There are a few differences in carrier proteins in some organisms. This is another example here of the electron transport chain. The Flavo proteins contain flavine from vitamin B2 or riboflavin to form FMN or flavin mononucleotide. This is the initial carrier molecule where NADH will drop off its electrons. This is one example of where a vitamin is required for a cell to derive energy from nutrients. The ubiquinones are non-protein carriers derived from vitamin K. This involves coenzyme A in the mitochondrion. The FADH2 drops off its electrons here. Because NADH passes electrons farther, generating more protons than FADH2, the NADH will result in 3 ATP, while FADH2 only results in 2 ATP. There are also metal-containing proteins, where the metals can alternate between reduced and oxidized states. Iron sulfide is used in various organisms, but copper is used in those with photosynthesis. There are also metal-containing proteins where the metals can alternate between reduced and oxidized states. Iron sulfide is used in various organisms that you can see here, and the iron goes between the states of iron 2 and iron 3. The carrier molecules can be diverse in bacteria, but the electron transport chain will always work on the same principle. The electron transport chain creates an ion gradient and uses chemiosmosis to generate ATP. With the protons moved to one side of the membrane, it has created potential energy we call the proton motive force. The cell uses the energy to propel down the electrochemical gradient through protein channels at the end called the ATPase. ATPase is the enzyme ATP synthase in a protein channel that catalyzes the phosphorylation reaction to add a phosphate to ADP to make ATP. It is referred to as oxidative phosphorylation because the proton gradient is created by oxidation of the components of the electron transport chain. And here we have an animation of the electron transport chain.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. The electrons, captured from donor molecules, are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain. NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved, ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Here we have another animation of the electron transport chain.
In the mitochondrion, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen. The terminal electron acceptor, and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Fermentation reactions will involve the partial oxidation of sugars or other metabolites. These will release energy using an organic molecule as an electron acceptor rather than the electron transport chain. Instead of passing electrons down the membrane, the cell oxidizes NADH to NAD+, and reduces organic molecules as the final electron acceptor. Microbes can produce a variety of fermentation products. A few examples are acetic acid or vinegar, ethanol or alcohol, or lactic acid. There are a few other pathways used to deal with nutrients other than sugars. In lipid catabolism, the enzyme lipase is used to hydrolyze the bonds attaching fatty acids to glycerol. The glycerol can join partway through glycolysis. The fatty acids will go through beta oxidation to degrade the fatty acids. Here, pairs of hydrogenated carbons are split off to make acetyl-CoA to enter the Krebs cycle. Because fatty acids can be several carbons long, several acetyl-CoA can be produced, requiring many turns of the Krebs cycle. Most cells will only catabolize proteins when the carbon from glucose or fat is not available. Humans will tend to spare fat when in starvation and catabolize protein. Some organisms, such as the food spoilers, will normally catabolize proteins. This is why certain foods, such as meat, have a tendency to spoil rather quickly. In deamination, the amine group is removed from the amino acid. It may be recycled, or the organism finds a way to remove the nitrogen waste. The proteases are the enzymes used to split the proteins into the individual amino acids. Here we're going to look at some of the major pathways.
In this chapter, you will learn about the pathways by which cells break down organic compounds and store the released energy in ATP. It all starts with glycolysis. All cells make ATP by capturing energy released during the breakdown of organic compounds, and all energy-releasing pathways start with the breakdown of glucose. In the cytoplasm, a metabolic pathway called glycolysis breaks glucose into two 3-carbon molecules of pyruvate. Energy from these reactions is used to make a small amount of ATP. Aerobic respiration continues in mitochondria, where pyruvate is broken down to carbon dioxide. The large amount of energy produced by these reactions is used to produce lots more ATP. In contrast, fermentation reactions continue in the cytoplasm. They produce no additional ATP beyond that from glycolysis. How the aerobic route ends. In aerobic respiration, pyruvate and NADH that formed during glycolysis enter a mitochondrion. Once inside, pyruvate is broken down to carbon dioxide by the reactions of the Krebs cycle. A bit more ATP forms and more coenzymes pick up hydrogen and electrons. In the final stage, electron transfer phosphorylation, coenzymes deliver electrons stripped from intermediates in earlier reactions to electron transfer chains. Movement of electrons through the chain supplies energy for the production of about 32 ATP. Oxygen is the final electron acceptor. Without it, aerobic respiration would grind to a halt. How fermentation routes end. Pyruvate from glycolysis can also enter fermentation pathways. These reactions are carried out in the cytoplasm, and they don't form any additional ATP. Fermentation reactions are anaerobic. They don't require oxygen. Instead, an organic substance is the final acceptor of the electrons originally derived from glucose. What cells do with food? Fats, complex carbohydrates, and proteins from food are broken down by the digestive system and then by individual cells into simpler compounds. These compounds become substrates for glycolysis or for the mitochondrial portions of the aerobic respiration pathway. Evolutionary Connections. Aerobic respiration breaks down organic compounds in the presence of oxygen to form carbon dioxide and water. This energy-releasing pathway arose only after the non-cyclic pathway of photosynthesis had added oxygen to the atmosphere. The two processes are now linked on a global scale. You may now click any topic to review an animation.